Okay, so we should be set here. We got to about, what, slide 10? Oh, 7. Went through this. And the only thing that I indicated on this slide was that we're going to talk about the actual metabolism of the amino acids. So we're going to talk about what happens when we remove the amine group and what happens to it and what happens to the rest of the carbons in that amino acid. Because in general, if we're consuming items that contain energy in our diet, those carbons in those molecules kind of have to be dealt with. They need to be metabolized. So we're going to look at what happens, for example, if we have too many of those amino acids, how they can store fat, how they can be used to make glucose, and other items. So that's kind of the intro to this. And then how we can make other nitrogen-containing molecules from them, because there's a lot of molecules in our body that contain nitrogen that we don't get in our diet. So we take amino acids, pull off the nitrogen, and use it to make nitrogen-containing molecules. So we'll talk about some nitrogen metabolism today. So how much protein is in the body? Protein is about 14 to 17% of the total mass of a lean adult. Now that doesn't mean that muscle tissue is 14 to 17% of the total mass, because that's a lot higher. Right? Muscle is about 60 or 70% water. This is just the protein. Okay, so it might not sound like a lot, but it's significant when you consider water is probably 65 or 70%, right? So for example, that's about 30 pounds for a 200-pound man. Skeletal muscle accounts for 40 to 60% of the body protein. In other words, most of our protein in our body is skeletal muscle. And the other major compartments are the skin, the protein in the blood and the skin. They're each about 15%. So that's about 30 plus for that. Those are the three main compartments. Okay, so when we need amino acids, it's likely they're coming from skeletal muscle. That's a major point of that. Have you ever heard that phrase, use it or lose it? I hate that more and more every day. But this is kind of what it means. It means that our body, including tissues that include amino acids and protein, dynamic. Constantly turning over. Even if you think about something like bone, that's a dynamic tissue. Muscle's even more so. So what this is showing is this is called protein turnover. It's basically how it's turning over every day. So for example, if you have a protein in the body and it's degraded, that liberates amino acids. Those amino acids have at least two main destinations. One is those amino acids themselves can be catabolized, and they're no longer amino acids. A second option is reuse the amino acids to make another protein. Depending on what's happening in the body of a person, the catabolic state, anabolic state, things that affect that kind of also direct what happens to those amino acids. Okay, but the point is it's consistently occurring. And that turnover is the dynamic balance between protein breakdown or catabolism and protein synthesis or anabolism. So this provides a means of adapting to conditions such as diet and stress. So for example, if a person has reduced calorie intake, so that's a diet effect, we might now break down more protein in order to try and get energy and glucose out of those amino acids. And the opposite is true, right? If we consume too many calories, we can actually add tissues in the body, and some of that might be skeletal muscle or nitrogen-containing molecules. Stress, a good way to look at stress, you could look at stress like it's week 10, right? Or anything that's stressful, physically or even psychologically, can enhance catabolism, and that can affect what's happening to this tissue. So in those conditions, you might have greater catabolism than anabolism. So we're kind of looking at this balance. And not many people realize this. We do. But let's say, for example, we go for a workout. Right after that workout, a lot of people are like, oh, I'm building muscle. You're actually not. You're actually breaking down muscle. You're in a catabolic phase. The only way to change that from a net catabolic to a net anabolic, diet. That's why we can immediately use nutrient intake to shift that, change the hormonal profile, change our nutrient assimilation so that now we change to anabolic phase. Okay? But anytime we're not stimulating skeletal muscle, it tends to go away. And as soon, for example, as soon as a person becomes bedridden, catabolic phase, through the roof. It's really problematic. That's why you always want to, if you can, during the recovery process, get them to stimulate their skeletal structure so that it helps them maintain lean body mass.
What did you say happens when we eat too many calories?
We can actually add tissues. So in that case, we can be more in anabolic phase and catabolic phase. So that's one of the things that changes that.
Part of like a sedentary lifestyle, kind of the same as being bedridden, because you're just not stimulating your muscles.
Yep. Yeah. So, like next week during grading, potentially catabolic for me, right? If you're not exercising and continually doing this, but if you think about the advantage of that, if I'm not using a particular muscle, I can use energy to add and maintain the muscle that I am using. So even if you change your workouts and you go, oh, I'm doing a lot of running this week, and the next week you go, I'm doing all upper body yoga stuff, then it can actually change your composition. And I think a lot of people don't realize how significant this turnover is. So I'm going to give you a number. It's estimated the amount of protein turnover is equal to the body weight in kilograms times 4.6. This is per, let's say 24 hours. So for example, if a person that weighs 70 kilograms, we love this, right? Approximately how many pounds is that? Probably 165 pounds, something like that. 70 kilograms times 4.6, you guys can do this in your head faster than I can. That's approximately 320 grams. In 24 hours, that's the amount of protein that's turned over in the body. Is that how much you eat? You guys all eat 320 grams a day? Probably not even close to that. So what's happening? 320 grams of protein in your body is basically being broken down and metabolized, right? So that's a lot more than we typically eat. But this much is turning over. So how do you explain that difference? 320 grams is being turned over, but we only need to eat maybe a fraction of that. If you do the math on this and you say this is a 70 kilogram person, approximately what's their recommended protein intake? What's that, 56? That's a big difference. So why is there that big difference?
We make a lot of it, like our bodies.
We reuse it. Reutilization. So put this in your notes. The most significant source of amino acids for protein synthesis are amino acids that are being reutilized. So another way of thinking about that is, what's the source? Endogenous amino acids. If we're not reutilizing those amino acids, let's say we're under a traumatic physiologic state, and some of those amino acids are being used for glucose or energy or just being broken down and being excreted, what does that do to our protein requirements? Dietary protein requirements go up. So that can be one of the factors on whether or not a person's getting adequate amounts of protein. Their ability to reutilize amino acids that have been liberated from another protein. Because if you think about it, let's say on Monday I've got a muscle fiber where an actin molecule has an amino acid in it. On Tuesday, that amino acid could be on a hepatic enzyme. Our body uses the amino acids. It can several times to make several different proteins. Okay, so the good thing of it is it decreases dietary protein requirements significantly. Even though we're adapting constantly, we can still reuse it. Okay, so now let's look at a little bit of this as review. Let's talk about digestion and absorption. Right, remember the digestion of a protein involves denaturation by hydrochloric acid and then hydrolysis by digestive enzymes. So hydrochloric acid denatures the proteins. So if this is a protein that we consume, there's two ways to denature. One is hydrochloric acid in our stomach. Another way is eating it, like when it's cooked. That denatures it. That's why sometimes its physical structure changes when we cook a protein. And now we have a denatured protein that now has open access to these peptide bonds for enzymes. It's kind of a two-step process, right? You can imagine enzymes not being able to get to some of the peptide bonds that are on the innermost parts of proteins. So the hydrochloric acid linearizes the protein to increase the efficiency of the digestive enzymes. Okay, so when we eat protein, here's some of the things that happen. Gastrin is released. Remember, that's stimulated by the presence of food in the stomach. Remember, gastrin stimulates hydrochloric acid production and release. That denatures the food protein, also activates pepsin. Remember, pepsinogen to pepsin. Remember, pepsin is an endopeptidase. So in the stomach, pepsin breaks down some of the protein. So you end up with some oligopeptides and even some free amino acids. So partial digestion of protein in the stomach. And this is a fun slide. So this shows the small intestine. So if you need to, you can say, lumen of the small intestine. We have an endocrine cell. It's basically right in the lining, that single cell thick layer that's lining the villi. We got an endocrine cells. We talked about that before. Here's an enterocyte, most common cell type. Right? This one releases hormones. This one absorbs molecules. Here they're kind of working together. On the surface of this enterocyte, there's an enzyme called enteropeptidase. It used to have another name, but this is a better name, right? Entero in the gut, and it's a peptidase, cleaves peptide bonds. So I'm going to abbreviate this for you guys, but look what's happening. When food goes into the gut, remember CCK is released. And we said a long time ago, and you remember this, CCK is the most significant effector of pancreatic exocrine function. So CCK stimulates the pancreas to release these. These are inactive enzymes, so we can call them zymogens. All of these guys, zymogens. Over here, they're active enzyme forms. So I want you to just remember one of these. enteropeptidase starts the process of converting zymogens to enzymes. Enteropeptidase starts the process of converting zymogens to active enzymes. Okay? So here's how it does it. Enteropeptidase converts trypsinogen to trypsin. And trypsin can convert the others. Enteropeptidase converts trypsinogen to trypsin. Trypsin converts the others. So this is fun with enzyme names, right? Here's trypsinogen. That's converted to trypsin. Chymotrypsinogen, converted to its active form, chymotrypsin. Proelastase, elastase, procarboxypeptidase A and B, 2-carboxypeptidase A and B. So the only names you need to remember from this, you need to remember that CCK stimulates the pancreas to release the zymogens. And then you need to remember trypsinogen converts to trypsin. Trypsin activates the others. And then enteropeptidase. Okay? So look what's happening though. Now you get all these enzymes that have been activated, and it all started with enteropeptidase. And enteropeptidase is there. It's on the surface of the intestinal cell. So as soon as trypsinogen comes into the lumen, enteropeptidase activates it, and now you get this cascade where all these proteases are activated. Happens every time you eat something. Get all these enzymes going into the small intestine, and a lot of them are proteases, aka peptidases.
So just to clarify, so in the stomach, it's like pepsin. Like once the pepsinogen is like converted to pepsin, that's like breaking down some of the protein, and then in the small intestine, it's mainly the enteropeptidase that's just like starting everything.
So in the stomach, you have pepsin that is really the only protease that's active in the stomach. Small intestine, you have all of these other enzymes that are secreted in their inactive form. And they're activated by trypsin, which is activated by enteropeptidase. So if you think about, again, why is the pancreas doing this? Because you don't want all these active proteases inside the pancreatic cells because they would degrade proteins there. So what do? Secrete them in an inactive form, and then they become active in the gut. It's the same thing with pepsin and pepsinogen, right? So it's pretty common. Now, why do you need all these enzymes? Because these enzymes, what are they looking for? They're looking for peptide bonds that are part of a specific amino acid sequence. Have you talked about this in any other classes? So one of the things I mentioned last time, I said, in a protein, you've got 20 different amino acid options at every part of that sequence. If enzymes are looking for specific peptide sequences and cleaving it, you need a lot of different enzymes to recognize all the potential amino acid sequences. And this is a very small list of all the proteases that are involved in this. But one of the points of this is proteins are relatively complicated to digest, but we can still digest significant amounts of protein and assimilate that. Okay? So now let's think about where we are. Dietary protein comes in, goes through the stomach, partial digestion, goes in the small intestine, significant digestion. That's where digestion and absorption are going to take place. So now one of the questions is, how is it absorbed? And I mentioned this like week 2 when we talked about digestive system a little bit. Okay? That's luminal protein digestion. There are also some peptidases on the surface of the cells. There's some that are called aminopeptidases and carboxypeptidases. What's the difference? Yeah, which end of the protein they work on. These are cleaving one amino acid at a time, either from the amino end or the carboxy end. So they're working on the ends of the proteins. So you end up with some free amino acids, and in here we just mean that free is single amino acids. And proteins that are getting smaller. So the question is, how small do they have to get for them to be absorbed? So what's happening is, along the enterocytes, we have transporters. And this is carrier, but that's not a great term. These are transporters that are in the brush border membrane. So in your notes, I'd put an asterisk by this and say, in general, amino acids need transporters to cross plasma membranes. Amino acids need transporters to cross plasma membranes. That's pretty much everywhere. Not only through enterocytes, but it's all cells in your body. Like if there's an amino acid in the blood, it needs transporters to get into cells. So that's a general rule in digestion and amino acid metabolism throughout the body. So let's look at two parts to this. The left side of this says, of all that dietary protein that's digested, approximately 33% of the amino acids absorb free form. In other words, single amino acids. So again, this should probably be better if it said amino acid. Right? Amino acid goes in. 67%, in other words, most protein is absorbed as di-tripeptides. That seems like a good idea, right? If we take in a lot of protein, it all has to be digested. I mean, cut us a break. Don't go all the way to single amino acids. Di and tripeptides work. So there's one main peptide transporter. It's called, and you don't have to remember that, so never mind. There's one main one that does this. Takes peptides, moves them in. Uses a proton as well. Okay? So this is a peptide transporter. So we absorb free amino acids, and di and tripeptides. Absorption into cells requires transporters of multiple types. We won't get into all of them. It's probably, we can spend weeks on that. All the different transporters. Some of them use sodium the same way as the SGLT1. Remember that? Sodium binds, increases affinity for substrate. So some of the transport is sodium gradient dependent. So it does require energy. So review the mechanisms of that. So does it take energy to absorb amino acids and protein? Yes. Just like glucose. There's a reason why we have that thermic effect of food, right? Where we say about 10% of the calories you use a day is basically absorption and assimilation. But here's the key. Even though we have a significant amount of di and tripeptides in that cell, they are hydrolyzed in the cell. They become free amino acids before they leave. So that's why this says peptidases. Basically break down those peptides to free amino acids. So their form going in the portal vein, free amino acids. Do they use the transporters? Yes. Now they're in the portal vein heading to the liver. Another point of this is, in general, we don't absorb proteins, right? Intact proteins are problematic. They do things like create an allergenic response. So our body tends to just absorb amino acids into the blood to minimize that response. If there's a problem with the gut and proteins are going through into the body, that can be a problem, and that's how we can develop some allergies and things like that. But in general, amino acids are what get into that portal vein. Right. There's lots of stuff to talk about, like live enzymes and all that stuff, right? Live enzymes, good for my enzymes aren't alive. We generally take proteins, break them down to amino acids. So the most important thing about the protein is what kind of amino acids does it have, not whether or not it's live or dead. It's all dead. Okay? So there's lots of things, misinformation about this whole process. Okay, so a little bit about details about what's happening to the amino acids in the intestinal cells. There's a lot of options for them, right? Once they go into the enterocyte, some of them are used to make proteins. Like apolipoproteins, like you could tell me an apolipoprotein right now that's made in enterocytes. It's on the tip of your tongue. Like B48. You need that for chylomicrons, right? So B48 would be here. New digestive enzymes like enteropeptidase. Hormones, like CCK, nitrogen-containing compounds like RNA and DNA. How do we get the nitrogen from the amino acids that just entered this cell in general? All right, so enterocytes use some of it. Another unique aspect about these amino acids is glutamine. Glutamine is an important energy source for enterocytes. And it is because it does this. Glutamine will go glutamine to glutamate. I should spell this one. What's this? Alpha-ketoglutarate. Where's that? Krebs. So enterocytes can use glutamine to fill Krebs cycle. And as they do that, they just pull off nitrogen. So glutamine actually is an important energy source for enterocytes. It also stimulates their growth. So glutamine is an energy source, and it stimulates growth of enterocytes. So later on, if you guys are prescribing any type of enteral nutrition plans for some patients, sometimes you'll put extra glutamine in there to help support the health of their gut or the recovery of their gut. It's important for enterocytes. So it's unique for that. And the enterocytes can actually also pull it from the blood. That's how important it is. So we'll talk more about glutamine coming up. Some of you might see it as a supplement. The recommendation to use it as a supplement, especially related to exercise, ambiguous. I'd say it's not really proven consistently to be effective. So that might be where you see it. Okay, so now, let's say we've gone from the portal vein to the liver. Now, as you can imagine, lots of options in the liver. So the liver is a major site of amino acid uptake following dietary intake. So the question is, what happens to all those amino acids? So we're going to talk about these 3 bullet points, but I want you to remember those percentages. 20% of the amino acids are used for making proteins and nitrogen-containing compounds. All right, so they are used by the liver to make hepatic proteins or other molecules that contain nitrogen. And I'll show you examples of this in a minute. The next one is maybe the most surprising. 57% of the amino acids catabolized. Almost 60% of that protein that I ate that got to the liver, the amino acids broken down. And so what that means is, let's say we have Thanks for reminding me. Here's a generic. The amino acid is broken apart. It's catabolized, the amino acid itself. What good does that do us? Provides carbon sources, nitrogen source. Does that seem high though? 60% of the amino acids that we eat, this happens to it. You can look at it like, well, that doesn't seem very effective. Or nitrogen must be really important, because the liver has the ability to pull lots of nitrogen from that protein. And we'll look at examples of that. About 23% of the amino acids go back into the blood without being metabolized. All right, so they kind of go through the liver, and then they go to other tissues. Okay, so after you eat, you will see an increase in amino acids in the blood. And then after a few hours, those amino acids go into tissues, and you see that go down. So let's focus on this first one for a couple slides. This is the synthesis of proteins and nitrogen-containing compounds within hepatocytes. Some of the things that are made. Albumin, remember albumin? We've talked about that a lot. Made in the liver, secreted into the blood, secretory pathway. A lot of hepatic enzymes. Some of the amino acids become other amino acids. We'll look at interconversions in a second. Purine and Pyrimidine Bases for DNA, for example. Those are nitrogen-containing. So we don't have to consume DNA in our diet. We make DNA from using nitrogen from dietary protein. And the last is creatine. Everyone's probably heard of that, right? Yes, we make it. It's not just at GNC or on bodybuilding.com. Okay, here's creatine. Here's its structure. Contains a lot of nitrogen. But look what happens to it. Creatine becomes creatine phosphate, also known as phosphocreatine. And look how it does it. As this name implies, it's phosphorylated. Sorry, these are just floating, but these are actually connected. Here's your phosphate. So why do cells do that? This is a high energy phosphate bond. So look what it does. When you go from the phosphorylated creatine back to creatine, you can make an ATP. Okay? And then what you can add to this slide is, when it goes from creatine to creatine phosphate or phosphocreatine, this is during rest. So where do you get the ATP? Krebs cycle, ETC, and then some of that ATP phosphorylates creatine. Over here, put high intensity work. High intensity work. So you can put in your notes. Right? This is, so creatine, the last step in creatine synthesis in the liver. But then what happens to it? The liver secretes it. So the liver makes creatine, secretes it, and 95% of your body's creatine is where? Skeletal muscle. 95% of your body's creatine is in skeletal muscle. And it serves it as an energy source during short duration, high intensity, anaerobic physical activity. So it can enhance strength and strength-related exercise performance. Sure. So creatine and creatine phosphate can enhance strength and strength-related performance. So if a person's doing something like an exercise or martial arts or football or baseball or things like that, during those high-intensity short bursts, in general, the more creatine phosphate in a muscle fiber, the longer you can do that high-intensity exercise. And one of the reasons is if you look at a muscle fiber and you measure the amount of ATP in there, it's actually very low. If you measure the amount of creatine phosphate, it's higher. So it's kind of a backup energy support. It's more stable than ATP. So you find a higher concentration. That way, as soon as that ATP is used, creatine phosphate can basically remake that ATP so you go a little bit longer. So depending on what a person's doing, it is helpful. So we do make it. So one of the questions is when we consume it, does it actually add more creatine to muscle fiber? In general, yes. Mm-hmm.
So is that why when people take creatine supplements, they say they have to drink more water because of the ATP that's going into it? And from EDC, how that needs water?
No, good thinking, but it's a little bit different. This is a charged molecule. So the more creatine and creatine phosphate you have in a muscle fiber, the more water it pulls. So it tends to hydrate the muscle. And in general, a hydrated muscle does better than a dehydrated one. Uh-huh.
Do people have to like cycle this because
Yeah, you really should. If excess amounts of creatine can cause problems like hepatic dysfunction. So I've known, I think, 2 people over the years that have been on creatine for a long time, so long they can't remember when they weren't on it. And if they do a liver enzyme panel, they'll see elevation. And one person came to me, I said, you need to stop that right away. Tell me what you're eating, blah, blah, blah. I said, what else? Oh, nothing. I said, what else? Creatine. How long have you been on it? Probably since I was 16. Right, it's not funny. And then they got off it for a while, and then the liver enzymes went down. You know, sometimes if you're looking for liver function, you measure enzymes that you only expect to see inside liver cells. If you see them go up in the blood, that means the liver cells are lysine and they're being damaged, and enzymes are escaping into the blood. So you can use that as a marker for hepatic dysfunction. We'll talk a lot about that in clinical. And a couple of those enzymes are related to amino acid metabolism because the liver is so important for amino acid metabolism. So my recommendation is people should be off it at least as long as they're on it. So a person wants to go on it for 2 months, all right, stay off at 3 if they're determined to go on it. Most of the studies indicate it's relatively safe, but still for college athletes, you can't give them creatine. They can take it, it's not banned, but it's prohibited. Meaning, sports programs can't give that to them, but if they're taking it, it's not illegal. And a lot of times the athletes gain weight because of the water. Like I know if I start a creatine cycle right now, probably within 2 or 3 weeks, I'd gain 8 to 10 pounds. Easy. And that's sometimes what keeps people on it. Because they know, you know, it's hard to understand, but for some people that drop in weight affects their psyche, their physical performance, their strength. But it can be effective. And there's lots of hocus pocus. Oh, this creatine's better. Not really. It's creatine monohydrate, none of the fancy forms. That's creatine. Okay? So, but there's a lot of money in that.
Okay, so phosphocreatine is the active form? Is that like the point of learning it?
Yeah.
Okay.
But this is the kind you take. Goes into the gut, it's absorbed. Then when it goes to the muscle, it can be phosphorylated. Yeah. You guys might get questions about that. That's why I spent a few minutes on that, right? This looks fun. Simple. A lot of things happening in the liver. I'll make a couple points to this, but this is some examples of amino acid metabolism in the liver. By far the most important metabolic site for amino acid and protein metabolism. But look what's happening, like even histidine, tryptophan, aspartic acid, glutamine. Those are the amino acid forms in the rectangles in the boxes, making other molecules that contain nitrogen. So one of the points of this is a lot of amino acids make other nitrogen-containing molecules. That's kind of what's going on at the top. Here's another point to this. Essential amino acids can be converted to non-essential amino acids. Top bullet point. Let's use this example. Phenylalanine, essential. Tyrosine, non-essential. Why? We make it from phenylalanine. But here's what doesn't happen. Non-essential amino acids don't become essential amino acids. Makes sense, right? One essential amino acid doesn't convert to another essential amino acid. In general, we don't make the essential amino acids or make them in adequate amounts. Okay, so lots of nitrogen-containing molecules made from those. Here's the last point. Some non-essential amino acids become conditionally essential in conditions where their synthesis is impaired. And if we look at the example that I just mentioned, phenylalanine becoming tyrosine, and you may have already learned how to draw those structures because those are the structures I expect you to remember. And I'm just going to go, here's phenylalanine. Here's tyrosine. That's the only difference. It's nice, right? But just do that and you know how to draw another one. So if you're going to name this enzyme, phenylalanine hydroxylase adds that. Unless something's wrong with this enzyme. Unless there's a genetic alteration in this gene that causes an abnormal structure to the enzyme and it becomes either ineffective completely or less effective than the person has PKU. And that means that what happens is these molecules accumulate. That's when phenylalanine accumulates and becomes phenylketones. So they're modified to ketone structures, and they end up in the blood and in the urine, and when they end up in tissues like the central nervous system, it damages them. It's harmful. So PKU is what's called an inborn error of metabolism. You ever heard that? That's a genetic alteration that affects a metabolic pathway. In this case, it's affecting the metabolism of phenylalanine. And so what's the most important tissue for this? The liver. So lots of labs, friends of mine worked in labs years ago that were working on gene therapy for PKU and trying to get enhanced expression in people for this enzyme in the liver to try to attenuate the negative effects, right? So these become toxic. It's pretty common. Probably right now, I'd guess there's 2, maybe 3 people at Cal Poly with PKU. It's about 1 in 14,000 or 1 in 16,000 in a typical population in the U.S. Some ethnic groups are higher because of the genetics and lower, but pretty common. A few years ago, we had a nutrition student. Her younger brother had PKU, and typically the parents know, so they know it's an autosomal recessive. So you can kind of follow the pattern, and if they're carriers, then they know the likelihood that one of their kids will have it. And they screen for it typically, and then they change their diet. So if you're going to change their diet, when would you do it? Immediately, right? And what would you do? So in newborns, what do you do?
Don't give them any phenylalanine.
Okay, don't give them any phenylalanine. You gotta have some. Gotta make proteins, right? This is one we don't make. So you control their phenylalanine intake. You give them a synthetic diet. It's a synthetic formula that has a set amount of phenylalanine. What do you do with this?
Some more.
Increase or decrease? Give them more. So you decrease phenylalanine, increase tyrosine. Because under this condition, this is now essential. Right, so newborns, you give them formula. What do you do as they grow? They can control it through diet. They'll tend to control plant-based versus animal-based proteins. Animals tend to have more phenylalanine than plants. So they'll adapt their diets. And in general, they're consistently monitoring blood for phenylalanine levels and phenylketones to make sure it's controlled. But if they control it, they can have perfectly normal lives. Just gotta watch what their diet. Phenylketonuria. Yeah, because the phenylketones end up in the urine. Got it?
What kind of side effects do the phenylketones have?
They'll actually damage neurons, so it can compromise mental development, coordination, quite a few things that involve the brain and central nervous system. Again, I mean, in the U.S., we, lots and lots of reasons we're very fortunate to be in a developed country. So in other parts of the world, it's a completely different scenario. Good here. I think that's the main points here. Feel better? Not too bad. Just a few overviews, a couple specifics. That's about it. So let's take our break here. Let's take like a 5-minute break, and then we'll finish this one and move on.